Thank you. And thank you for inviting me and letting me be part of this symposium. And I should specifically thank Nakamura-san for inviting me. Actually, I'm a physical electrochemist, and before this morning, I never actually thought about the origin of life. So I'm not sure if I will have anything to tell you about the origin of life. But yeah, if you don't find this useful, you can blame Nakamura-san afterwards. So I work in the area of electric catalysis, and this is what Nakamura-san asked me to talk about. And specifically, a part of my group works on the electric catalysis of CO2 reduction, and the relation with proton-coupled electron transfer. And maybe it's useful to explain a little bit about how an electrochemist looks at redox reactions, or how we study them. So the way I study redox reactions in an electrochemical cell is that I have an electrode, and the electrode is typically also the catalyst for my reaction, and I apply a potential using basically an electronic feedback piece of equipment called a potentiostat. And with that piece of equipment, I can change the electrode potential of my electrode from reducing, which would be a very negative potential, to oxidizing, which would be a very positive potential. And then I measure the current that is flowing through that electrode, and that current gives me the rate of the reaction. And in that way, I can generate current voltage curves of redox reactions. And then if I study a particular redox reaction, obviously I will need to know where let's say the redox potential, or the equilibrium redox potential of that redox reaction is. And then typically if I would reduce CO2, I would ideally see some current flowing, and reduction current in electrochemistry we consider as a negative current flowing. As I make the potential more negative, I would expect some reduction current to start flowing, and then this current potential curve would typically look something like this. And this is then what I measure electrochemically. And if I reduce CO2, then I can make a number of different products, and we've seen some of those already today. And obviously from the current, I can't really tell what product I'm making. I can just tell how many electrons are flowing in the external circuit. And then I have a number of important things that I look at. I mean, typically we would like this to happen very close to the redox potential, because my interest is actually not so much in the origin of life, but my interest is in seeing if I can use CO2 and water and renewable electricity to make interesting molecules, and I would like to do that as efficiently as possible. And that means that I would like this to happen as close as possible to the equilibrium potential. Now, this typically doesn't happen, and it means that in order to get some kind of current, we need to move the potential away from the standard redox potential. We call that an overpotential. And if that overpotential is very small, then basically I have a very good catalyst. And if that overpotential is very large, then basically I have a very bad catalyst. And much of what we do in electrocatalysis is to find electrode materials that make this overpotential very, very small. But obviously I also need to worry about the product that I make. So I also need to worry about the selectivity. Now, if I would not do this electrochemically, but I need some other kind of voltage source, I would probably use another redox couple to basically donate the electrons to reduce CO2, and that would be let's say the other redox couple. Hydrogen would be a good example. And obviously this redox couple needs to have a redox potential that is more negative than the redox potential of the species that I'm trying to reduce. And then in corrosion theory, we define that we make this mixed potential where the current of the oxidation reaction and the current of the reduction reaction is equal, and then we end up at this sort of mixed potential where everything happens. Now, before I move on to trying to explain how these reactions take place and how I can make good catalysts for this, and what is the role of protons and electrons, there's one thing that I included as a slide because of the talk that we heard this morning about oscillations, because that actually turns out to be my previous life. I did a PhD in studying electrochemical oscillations and instabilities. And typically what you would expect if you apply a fixed potential, that after a while you would expect to get a constant current, a steady state current. Or if you apply, and this is something that we can do also with our feedback electronics, we can apply a constant current, and they would expect that the system develops a stable potential. But this very often doesn't happen. And this typically what we see is sometimes that when these curves do not look sigmoidal like this, but develop a branch where we have a negative resistance or a negative impedance, then we can develop instabilities. And these instabilities can give rise to oscillations. And I show you a few here. And these are really relatively simple reactions. This is the reduction of indium 3 plus on a mercury electrode. And I will not go into the details, but this was a question I think that was asked this morning. If you can develop something like chaos, here you see the current oscillate in the reduction of indium on a mercury electrode. And this is how we drew what is called a strange attractor from these oscillations. You can see here a return map. I don't know if you're familiar with this kind of terminology, but what we see here is a deterministically chaotic oscillation in the reduction of indium 3 plus on a mercury electrode. And this develops through a series of period doubling bifurcations. And you can have very complicated oscillations. Here we see something that people call bursting oscillations. This is the reduction of hydrogen peroxide on a platinum electrode. And now we drive this as a constant current and we measure the voltage. And you see that it seems to go through two different types of oscillations, one with a very high frequency and one with a lower frequency. And I don't want to dwell on this, but this really, much of this can be understood simply from the current voltage curve, where we have a region of negative impedance that is coupled to the external circuit and that is coupled to a slow feedback mechanism, which is very important in these type of instabilities. And in this case, the slow feedback mechanism is mass transport. And actually, we showed in a very simple model here many years ago, and this was during my PhD, that we can understand much of what is happening in this system by simply coupling the kinetics that give rise to the negative impedance to mass transport. And that will give you rise to oscillations, instabilities, and all the transitions to chaos that we see. So the chemistry behind that is relatively simple. What we have here is a very simple nonlinearity that is coupled to a slow feedback mechanism, and that can give rise to these type of instabilities. But that is not what I want to talk about. What I want to talk about is about how to catalyze electron transfer reactions. And one thing you may have noticed, if we catalyze CO2 to, let's say, CO or formic acid or methane or methanol or ethanol, and you will see that these are things that we can make, that this requires a different number of electrons. And one thing I've learned is that the overpotential that we typically find to catalyze a certain reaction depends on the number of electrons that we're trying to transfer. If we transfer a single electron, then this is something we would model using a theory that you may have heard of, is the Marcus theory for electron transfer. And the Marcus theory of electron transfer tells us that the rate of an electron transfer event depends on how easy or how difficult it is to reorganize the environment of the redox center, basically the solvent. If it is very difficult to reorganize the solvent, then the rate of electron transfer will be slow. If it is easy, the rate of electron transfer will be fast. Basically what it is saying is that the solvent reorganization or the environmental reorganization of the redox center is your reaction coordinate that basically accommodates the electron transfer. Once we transfer 2 electrons, things are a little bit more complicated, or maybe I should say easier, because one of the things that we find from Marcus theory is it's very unlikely that you transfer 2 electrons exactly simultaneously. And that is because the solvent basically needs to reorganize too much. And it is practically always more efficient to transfer these electrons one by one. So we transfer the first electron, then we generate an intermediate, and then we transfer the second electron. And once we have to catalyze a 2-electron transfer reaction, what the catalyst does, it basically allows for the intermediate to exist, and it gives this intermediate, it generates an intermediate of the right energy. And this optimization of this intermediate is really the key to having a good catalyst. And if you get just the right energy of this intermediate, and I'll show you in a minute what I mean by just the right energy, we can reach a situation where we have very low overpotential. Let's say effectively almost zero. And we come to the conclusion that it is in principle possible to catalyze 2-electron transfer reactions with the right catalyst in a way that there is hardly any overpotential. That also means that that catalyst is capable of catalyzing both the reduction and the oxidation, so in both ways. I will refer to that as reversible catalysis. Once we transfer more than 2 electrons, you could, for instance, the oxidation of water to oxygen, the oxygen evolution reaction is a good example. It requires 4 electrons or 6. What we see then is that we no longer have a single intermediate. We go through multiple intermediates in our catalytic scheme. And now we need to optimize every single intermediate to get our optimal catalyst. And the problem here is those intermediates are similar and they bind to the catalyst in a very similar way. That means if we bind one stronger, we will bind the other intermediates stronger as well. And that means that we cannot optimize all these intermediates independently. They are fixed through what we call scaling relations. And these scaling relations make it very difficult to reach a situation where you can have non-zero overpotential. And that typically means that 4 electron or 6 electron transfer reactions, although they can be catalyzed by a single catalyst, they practically always have an overpotential. And it's very difficult to overcome. This is one of the reasons why it is difficult to do water splitting and why it is difficult to develop fuel cells in a fully efficient way, because we have a 4 electron transfer reaction there that is very difficult to catalyze because it has more than one intermediate. And you will see that these ideas also apply to CO2 reduction. Just the same thing, but now in a way that maybe you recognize from the Marcus theory. In the Marcus theory, we have these different oxidation states of the system. We draw the free energy as a function of the reaction coordinate. We have these parabolas. This is the initial state of the system. This is the final state. And this is our intermediate state. Now you can see that if we try to transfer 2 electrons simultaneously, the activation energy here is going to be the intersection of these 2 parabolas. It's going to be somewhere here. So that activation energy is very high because we need to reorganize the environment too much. So it makes sense to find this intermediate oxidation state to generate this extra minimum here in the potential energy landscape. And now here you see the activation energy of the separate steps. And now what determines a good catalyst is not so much actually the activation energy that we have here, but it is the level of this intermediate energy. It can be high, it can be low. And what I'm going to argue with is that the best catalyst will actually will place this energy level here exactly at the same energy as the initial and the final state. So we generate this flat thermodynamic landscape. That is going to be our optimal catalyst. And that is basically just an expression of a principle that is known in heterogeneous catalysis as the Sabatier principle. The catalyst should not bind this intermediate too weakly, it should not bind it too strongly, it should bind it at an intermediate energy level. That basically means that close to equilibrium, the energy of the intermediate has to be equal to the energy of the initial and the final state. And so that gives us a very clear quantitative argument of how to find the best catalyst. Basically, we just need to find a catalyst that binds that intermediate with just the right energy. And nowadays it's possible to calculate energies of intermediates using quantum chemistry, using density functional theory calculations, for instance, and that will allow you to screen many different catalysts, and this is a way to search, using a computer to search for a good catalyst. And this is a strategy that is used very much nowadays in heterogeneous catalysis and also in electrocatalysis. Now, other than that we're transferring electrons, we're also transferring protons, and this brings another reaction coordinate. So what I'm assuming in many cases that when you're transferring a proton and an electron, and what many people assume in the literature is when you're transferring both a proton and an electron, that this happens concertedly. So we transfer a proton and an electron concertedly, so we go from A to AH. And so to understand the thermodynamics of this pathway, we just need to know the energy of this initial state and this final state. But you could imagine that it's possible to have pathways where you first transfer an electron and then transfer a proton. And this I will refer to as decoupled proton-electron transfer. Now, there's many theories on this, beautiful quantum mechanical theories, but I think the real issue in understanding whether you have concerted proton-electron transfer or whether you have this decoupled proton-electron transfer just follows from very simple thermodynamic considerations. And that just has to do with the stability of these 4 states. Typically, if these 2 states here are high in energy, you could imagine that you will have concerted proton-electron transfer simply because these 2 states are energetically unfavorable. But once these states are favorable, once this A likes to pick up an electron, for instance, because it has a favorable electron affinity and it has a good solvation energy, then you can imagine that you first transfer an electron and then a proton. And you can make things more complicated. We can start transferring 2 protons and 2 electrons, and we get these kind of square schemes. And typically what people assume in the literature, that things go along this diagonal. But what I'll argue is that in many cases, actually, the mechanism follows something like this, where somewhere in the mechanism we decouple proton-electron transfer, and we follow a pathway like this. Now, how does that manifest in an experiment? Well, if I apply again the Sabatier principle, the idea that every step here needs to be close to equilibrium in order to have the optimal rate, and if I look at this step here, this is just an acid-base equilibrium. And if I want the acid-base equilibrium to be close to equilibrium, that basically means that I want the pH of my solution to be close to the pKa of this acid-base equilibrium. Now that suggests that when you have decoupled proton-electron transfer in your mechanism, there's going to be an optimal pH at which the reaction likes to take place. And this very simple idea explains a lot of data in the electrocatalysis literature. So what I'm saying is that once I have this decoupled proton-electron transfer in my mechanism, there's going to be an optimal pH. And so if I look at the rate at a fixed thermodynamic driving force, I should say that this rate is at the same thermodynamic driving force for every pH. It's very important to take that into account. Then there's going to be an optimal pH, and that optimal pH is going to be very close to the pKa of the acid-base equilibrium in the mechanism. This explains why many alcohol oxidations prefer to take place in alkaline media, because alcohols deprotonate in alkaline media. They have a pKa of around 14 or 15, and the deprotonated alcohol is more easy to oxidize than the protonated alcohol. Nitrate reduction prefers to take place in acid media. Nitrate has a pKa of -1 and actually prefers to be protonated before it reacts further. And that's why nitrate prefers to take place in very acidic media. An intermediate case is formic acid oxidation. Formic acid has a pKa of 4, and we find that if we want to optimize the rate of formic acid oxidation, for instance in a formic acid fuel cell, the pH of the electrolyte should be close to 4. That doesn't necessarily optimize the conductivity of the electrolyte, but it optimizes the rate of the catalytic reaction, of the formic acid oxidation. So what I want to do in the rest of the talk is show some of these principles in relation to the electrocatalytic CO2 reduction. If you look in the literature, in purely the electrochemistry literature of CO2 reduction, there's basically 3 major products that we observe. CO, formic acid, we've seen them already as the initial reduction products of CO2, and oxalate. Oxalate is typically a product in a-product solvents. And all these reactions are 2 electron transfer reactions, which I think is not a coincidence. These are relatively easy to catalyze. And actually, there are catalysts that catalyze these reactions reversibly. I'll show you an example in the next slide. Enzymes, there exist enzymes that can catalyze these reactions reversibly. So they can catalyze them in both directions with essentially zero overpotential. If you want to reduce them further, then these are the intermediates, so you can reduce CO further to methane, ethylene, and other interesting products on copper electrodes. I'll show you that in a minute. And you can also imagine that you oxidize further this carboxylate functionality to an aldehyde and eventually to an alcohol. Now, just to show you that you can do this reversibly, so you can reversibly convert CO2 into formic acid in this case and back. Here's an example of an experiment from the group of Judy Hurst in Cambridge, where they immobilized the formate dehydrogenase on a pyrolytic graphite electrode. And they basically, they changed the potential at which the electrons are transferred to the active site. In this kind of protein film voltammetry. Here is the redox potential of the conversion of formate to CO2. If you go negative, you see that you reduce CO2 to formate, and if you go back, you see that you oxidize formate back into CO2. And note that this happens essentially reversibly around the equilibrium potential. showing that this 2-electron transfer reduction with the right catalyst, this 2-electron transfer reaction, can be catalyzed reversibly. And this inspired me to see if we can also do this with something that is not an enzyme. And this comes the closest to what we find so far. This is the reduction of CO2 on a platinum palladium catalyst. And basically what we did, there's a lot of literature, electrochemistry literature on formic acid oxidation. And the best catalyst that people found was a mixture of palladium and platinum. We just took that catalyst and we ran it in reverse. And we found that we can reduce CO2 to formate with this catalyst. And we can do this, almost quasi-reversibly. And you can see that very close to the equilibrium potential, you start generating formate. This is not a very stable catalyst. It generates also CO, and CO will poison the electrode. But this is just to illustrate the principle. Here's another example of a system that we studied. This is a cobalt porphyrin or protoporphyrin that we immobilized on a graphite electrode. We actually initially we were interested also in using this catalyst for nitrate reduction, but at some point we decided to see if this could be active for CO2 reduction. We follow this system with an online mass spectrometer. Here you see the negative current flowing as we make the potential more negative. And actually, I was expecting to see CO because this system had been studied already, and it was found to make CO. But we find actually that this specific system can make methane, so it can do an 8-electron transfer reduction from CO2 to methane. The selectivity, however, is not heartwarming. This is about 1%, and 99% is hydrogen, because all this is taking place in water, and water is also reduced by this catalyst to form hydrogen. But the interesting thing here is that this reaction is extremely sensitive to pH. If we shift from pH 1 to pH 3, we see that we shift the formation of hydrogen to more negative potentials, but the reduction of CO2 still takes place at the same potential. And we generate methane, and now we can also see the formation of CO in our mass spectrometer. And now we find that the formation of CO at pH 3 can reach selectivities of up to 60%. And that of hydrogen is much lower. Now, there's different ways of looking at this, but the key reason why I think that this is so sensitive to pH is that the hydrogen evolution reaction is a reaction that takes place through a concerted proton-electron transfer and is not very sensitive to pH. But the CO2 activation is a process that takes place through an initial electron transfer. So we start from the cobalt porphyrin here. If we reduce that from the 2+ to the 1+ state, that is when the CO2 binds to the cobalt center. And this is just an electron transfer. And the protons are only transferred later. So here we decouple proton from electron transfer. And that makes this cycle pH sensitive and this cycle not. And so now by playing with pH, we can optimize not only activity, but we can also optimize selectivity. And we can tune the pathway that we want to take place. Now, we've also studied, we can actually calculate this. I don't really want to dwell on this very much. We actually played with the metal center of these porphyrins to see if that has an effect on the product that we make. And we find that typically iron and cobalt tend to make CO. And we find that rhodium, indium, and tin, interestingly, make formate. They make a completely different product. And we wanted to understand what property of the catalyst it is that determines this selectivity. And the classical theory in this area is that has to do with the way you make the first intermediate and how that first intermediate binds to the catalyst. If you bind that first intermediate through the carbon, so you have a proton-coupled electron transfer to the CO2, and you make this COOH intermediate that binds through the carbon, then in the next step you will break a CO bond and you will end up with CO. But if you transfer that first proton-electron transfer to the carbon here and you bind through the oxygen, then you make a kind of formate intermediate, and this will dissociate from the catalyst and your product will be formic acid. And all this is always in competition with hydrogen evolution. And this is something, again, that you can calculate. You can do DFT calculations for all your different catalysts, and you can see which intermediate binds the strongest or is binding the most favorable to the catalyst. And in that way, you can predict what kind of product you will make using quantum chemical calculation. Now, this did not allow us to explain our data on the different porphyrins, and so we came up with a different mechanism. We actually found that if we do this on indium or tin, CO2 does not want to bind to the metal center. And we also find that we do not reduce the metal center of indium and tin porphyrins. What we reduce is the ligand. And we generate a hydrogen, or actually we generate a hydride on the ligand. We generate an H- on the ligand, and this H- attacks the carbon of the CO2, and this generates the formate. And we find that we can generate that H-, that hydride intermediate, not only on the ligand, we find that on indium and tin protoporphyrins, we make it on the ligand. On rhodium, actually, we find we can make it on the metal center. And so rhodium is activated not by binding CO2, but rhodium is activated by binding a hydride, and this hydride attacks then the CO2, and that is the way in which you make formate. So our idea of how the selectivity of this reaction is determined is a little bit different from some of the ideas that exist in the literature, and we think it is really the nature of this nucleophilic attack that you have after you've reduced the catalyst that determines the product that you make. If the electron density is located on the metal center, you will bind through the carbon and you will make CO. If the electron density generates a hydride intermediate, that hydride will attack the carbon and you will make formate. Now, we can reduce these products further, and one metal that is very good in that is copper, and this was actually this was discovered in Japan, not so far from here, at the University of Chiba, by Yoshio Hori about 30 years ago. And he discovered that copper electrodes, room temperature, aqueous media, are able to reduce CO2 and CO to what I would call non-trivial products like methane and especially ethylene here. And also ethanol is interesting. So we make a carbon-carbon bond electrochemically on copper electrodes. And we studied this again with an online mass spectrometry system. So here we can follow the formation of the gaseous products as we change the potential. So again, here we start from 0 volt. We go more negative. So we make the environment more reductive. And you see the current flowing here. This is, by the way, mainly hydrogen formation, as you can see here. You see that as we go more negative from CO2, we make some formic acid, but formic acid is not reduced further on copper. But what you can also see is that both from CO2 and CO, we make methane and we make ethylene. And you can see that these look very, very similar, these profiles that we measure in the mass spectrometer, suggesting that CO is an intermediate in the CO2 reduction. So we go from CO2 to CO, and then we reduce the CO further to methane and ethylene. And especially the formation of ethylene is intriguing. So how is this carbon-carbon bond made? And we studied this on this same reaction on single crystals. So now we play with the structure of the copper catalyst. Here we start from CO because that allows us to start playing with pH a little bit more than when we would use CO2, because CO2 itself is involved in all kinds of pH-dependent equilibria. If we do this reaction on copper 111, so where the surface atoms are arranged in this hexagonal fashion, you can see that we make methane and we make ethylene. You can see the profiles in the mass spectrometer are very, very similar, suggesting that they are made in the same pathway. If we do the same reaction, but now on copper 100, here the atoms are arranged in a square fashion, you see that you have a potential window where we only make ethylene, but we make no methane. So apparently here, the carbon-carbon bond formation happens first, even though thermodynamically speaking, the most favorable product is methane. So apparently this carbon-carbon bond formation is extremely sensitive to the structure of the surface. It is also extremely sensitive to pH. Note that here I compare pH 7 and pH 13. If I go from pH 7 to 13, note that the formation of ethylene shifts to less negative potential, so it happens earlier, whereas the formation of hydrogen still happens at the same potential. So apparently by playing with pH, again, I can play with the selectivity of the reaction. And again, I think this is because the formation of ethylene happens through a decoupled proton-electron transfer that favors this reaction in very alkaline media. The more alkaline, the better, and the higher the selectivity towards ethylene. To quickly take you through the mechanism that we think is happening. So we start from CO2, we make formic acid, but that is not reduced any further. We make CO that binds to the catalyst, and the reason why copper is such a good catalyst under these conditions for CO2 reduction is that it is the only metal that binds CO with an intermediate binding strength. All other metals either bind it too weakly or bind it so strongly that they become poisoned. For instance, platinum can reduce CO2 to CO, but CO binds so strongly to platinum that it cannot reduce it any further. This CO can be further hydrogenated. You make these type of intermediates. The CO bond will break at some point. You will end up with methane. Some of these intermediates may dimerize and form ethylene. And this is the pathway that happens on copper 111, and the rate determining step here is a concerted proton-electron transfer. But on copper 100, something else happens. Remember that there we make only C2 and we don't make any C1 products, at least in a certain potential window. And the way we explain this is through this dimerization mechanism. So we make a negatively charged dimer that binds to the surface, and this negatively charged dimer is the key intermediate, and that is then further hydrogenated, and this is made through an electron transfer step that is decoupled from proton transfer. And this eventually ends up in ethylene, but it can also generate ethanol in a bifurcation that happens somewhere here later on in the pathway. So what happens here is a kind of reductive dimerization or reductive CO coupling. My organic chemistry colleagues tell me this is very similar to McMurray coupling. There's various arguments that I can give why I think this is a reasonable pathway. One of them is that we've calculated this by DFT calculations, and we find that actually in the absence of solvent, I should say, the most stable configuration of the dimer is like this. And we indeed find that it's negatively charged. So if you put this kind of dimer on a copper 100 surface in a quantum chemical calculation, you will find that there's an electron transfer flowing from the copper into the dimer. And if you add solvent, the structure looks a little bit different, but it's still negatively charged, and it actually stabilizes because it is negatively charged. The solvent actually helps stabilizing this charged intermediate. And through that, we come to the conclusion that this is a very reasonable pathway. Interestingly, this can also explain why it is so sensitive to the structure, because to bind this intermediate to the structure, we find that we need these kind of sites on the surface. This dimer really likes to sit on these type of square sites. It doesn't like to sit on a surface where we do not have these square sites. And that explains why it is so sensitive to the structure of the catalyst and why it prefers 100 surfaces. Many people, when I show this, always ask me, do you have any evidence for this intermediate? Because this is just in DFT calculations. Can you see it using spectroscopy maybe? Well, maybe we do. These are some data that we generated using in situ infrared spectroscopy, where we find that very close to the onset of ethylene formation, on copper 100, we measure these infrared spectra, and we see two CO bands. One seems to be a single CO bond. The other seems to be a double CO bond. We only see this on copper 100. We never see this on copper 111. And when we try to assign these bands, we find that it doesn't correspond to anything that we can measure. And the only thing it seems to correspond to when we do DFT calculations is this type of hydrogenated dimer here that has CO frequencies that are relatively close to what we measure experimentally. So this may be considered as at least some kind of evidence for the formation of a key intermediate in the CC coupling on copper 100. Now, this seems a bit like an odd mechanism, this CO dimerization. But interestingly, nature does something very, very similar, not with CO, but with NO. Nitric oxide reductase is an enzyme that catalyzes the reduction of NO to nitrous oxide. And there are various mechanisms for this. I am not up to date about the mechanisms. As you can see, this is a very old paper. But one of the suggestions in this paper is that this enzyme catalyzes the reduction of NO by the formation of this NO dimer. And only after the formation of the NO dimer, you break the NO bond and you end up with N2O. So again here, NO reductase seems to have developed a reductive dimerization pathway at these low temperatures to make NN bonds. And what I'm suggesting is that for CO, you can have something similar, And the interesting experiment that we're trying to do now in my lab is to see if we use mixtures of CO and NO, if we can make CN bonds in this way. So far the results have not been positive, but we may have to tweak. We have to tweak the conditions a little bit. A final thing that I wanted to show is that actually, because these intermediates in these catalytic pathways are, in this case, negatively charged, they are extremely sensitive to the nature of the electrolyte, not only to the pH, but also to the cations that exist in the electrolyte. And this was actually already observed by Hori many years ago, that the selectivity to ethylene is very, very sensitive to the nature of the cation in the electrolyte, and we find that we see an enhancement in the formation of ethylene when we do this in cesium hydroxide, whereas in lithium hydroxide we see much less ethylene formation. And interestingly, in cesium hydroxide we also start seeing C3 products. And again, there's a bit of a debate in the community right now how to explain this. We have done some DFT calculations that suggest that the presence of cesium seems to have a positive effect on the stability of some of the intermediates. It acts like a kind of promoter. It sort of flattens out the catalytic landscape. But there are other theories that suggest that this has an effect on the local pH, and that may have then an effect on the final selectivity. The formation of C3 is something that is also interesting. I think this happens through a kind of CO insertion mechanism. It has been found that if you mix ethylene and CO electrochemically, and you reduce that electrochemically, you can make propionaldehyde, and that can be reduced further to propanol. So it looks like under these conditions, again, electrochemical conditions, aqueous solutions, we have some kind of electrohydroformylation reactions that can take place that allow us to grow these carbon chains. The final thing is that when this takes place, this always happens simultaneously with hydrogen evolution. That means that locally the pH is increasing, so any formaldehyde or aldehydes that we make will take part in disproportionation reactions, and we typically see that when we see alcohols, we also see carboxylic acids. So when we see ethanol, we almost always also see acetate, which suggests that actually what we make is acetaldehyde that is disproportionates due to the high local pH into the alcohol and the carboxylic acid or carboxylate. Okay, that brings me to the final slide. I showed you that it's possible to reduce CO2 close to the thermodynamic potential, but you should restrict yourself to 2 electron transfer, and then you can find the optimal intermediate if you have the right catalyst. We try to argue that the activation to CO or formic acid follows different pathways depending on the nature of the nucleophilic attack. That under electrochemical conditions we make these charged intermediates, and that means that these reactions are very, very sensitive to pH, and they're sensitive to the nature of the electrolyte, and specifically the kind of cations that you have in solution, and that you have somewhat unusual carbon-carbon bond formation pathways like reductive dimerization, and maybe electrohydroformylation is not so unusual, but it's not always considered in the mechanisms that people have suggested. Final slide, these are the PhD students, postdocs that were involved in this work, and the funding agencies, and I would like to thank you for your attention.
Okay, questions? I don't know if I should throw it that far. Might bonk somebody in the head.
Sorry, just to ask the obvious question. You made us aware that this is not your field, but I would like to know what you think about the situation 4 billion years ago, 4.2 billion years ago when life was starting. How do you see this happening? So many of us would like to see CO2 being fixed. Some people don't like it. Some people say molecules came from space. Some people say they did come from here, but not from CO2. How do you see it? So what would be your pointers for us in those of us trying to make molecules from zero, from very close to zero?
Wow, that's it. I think this should be considered. That's all I can say. I mean, it's very difficult to answer that question. I'm just pointing out that there are many different types of catalysts. One of the things I was thinking about this morning, what if you have small metal nanoparticles. I mean, that could act as catalysts as well, so not just ions. There are many different pathways out there. That's I think, my main idea, and it's very difficult to choose which ones would have played a role. Maybe they've all played a role somehow. I think the key really is how did all this organize into something like a cell? That is a completely different question.
Thank you very much. Great talk. So I think emergence of selectivity of catalyst is one of the key topics of prevalent chemistry. And you clearly demonstrated if we can control decoupling and coupling electron transfer, just change the pH, we can control selectivity. That's the point you talk, right, in the first topics. And my question is...
I think selectivity depends on many things. It depends on having the right catalyst, but also having the right catalytic conditions like pH, but also electrolyte. What I'm trying to say, I guess, is that the issue of selectivity is a very complex one and is one that you can tune in many different ways. And we're not always considering all these different ways.
Okay, so question is, can you rationally predict, so suppose if you have this kind of material, with that experiment, can you predict, this material have a function to decouple or couple?
Yes, you can predict if you know the pKa's of the different intermediates involved, then you can say which ones should take place at say neutral pH conditions, and which ones are less likely as a pathway.
Okay.
Hey, thanks for that. At some point you mentioned the hydrogen evolution reaction and how it generates pH gradients that create localized alkaline environments. One of the key types of mechanisms in biochemistry is the formation of an enolate, which then goes on to carboxylate something, which can form a CC bond that way. I'm wondering, how powerful is that, or how fast does that hydrogen release reaction have to be so that you could build up a localized alkaline environment strong enough to form an enolate from a ketone, for instance? Is that feasible, or is diffusion so fast that you can never form such a strongly alkaline environment? And how can I answer that if you don't know the question?
I would need to know the local current density,
Okay.
which is something that's not so, but it really depends on the rate with which you generate hydrogen and the rate with which you can replenish the proton donor. And if those are sufficiently different, you will have pH gradients. But those pH gradients also depend on the local buffer capacity. If the local buffer capacity is poor, then the pH gradient will be bigger as well. That's another parameter, actually. Buffer capacity can have a huge influence here on the selectivity of the reaction, simply by the fact that it generates different pH gradients.
Okay, one last question while Ryuhei gets set up for the next talk.
Mark, let me say I think that was the most rational talk I've ever heard on this subject. I mean, it was fantastic. What happens if you use, let's say, liquid ammonia, where you can actually inject an electron and have it sit in the solution, or HMPA? What do you see then?
Actually, if you reduce CO in liquid ammonia, you make a CO dimer. That is one of the reasons why I think the CO dimer is a reasonable intermediate.
What about CO2?
Basically, you avoid protonation reactions.
Right. But what about CO2? Can you make oxalate dianion?
Yeah, oxalate will be the product.
I should have had you chat with a graduate student who spent 6 months spraying ammonia all over everything, trying.
That would solve the ammonia problem, though, wouldn't it? Very good. Okay, so let's move on to the next talk, which will be given by. Actually, let's thank Marc again for it.